My great pleasure to welcome Carl Moth with us today. So Carl studied in Princeton, right?
Yeah.
You did your undergrad in Princeton physics, and then you moved for a PhD to UPenn.
Yeah.
So it's the University of Pennsylvania. And then you did actually, I think, two postdocs, right? One in Cambridge. So this was the time you moved to Europe.
Yeah.
And then for the first time you moved to Europe. And then the second time, and then you moved back to the US, to the Rockefeller University in New York City. It's also where we know each other from, to the Physics Center. And there you already started, I know of your work there, where you started to look at all the spatial networks together with Christoph, like in the vascular networks of the brain. And then you also started to think about more brain networks with Marcelo. There's this, I think it's a current biology.
Yeah.
Paper on this. And then since 2017, I think, you moved to Dresden in Germany, where you started your own group at the CBG, which is the Max Planck Institute for Cell Biology. Oh, is it cell biology genomics?
Genetics.
Genetics.
Yeah.
Genetics. And it's always fun to Center for Systems Biology, and then D is for Dresden, I guess, or Dynamics.
It's interesting.
It's fantastic.
They're just funny because of the address, it's always CSBD Dresden, so it's actually double Dresden.
So one theme I think that goes through your work is to use physics and also math to now address certain biological problems. You started off in the soft matter field, where you also looked at programmable shapes, so how we can actually change or induce also different shapes or change shapes, which is of course something also very important in development of organisms, because how an organism starts is like very spherical, right? And then there must be a lot of some of organisms, I don't know all organisms, but humans usually start very spherical, and then there must be a lot of shape changes going on, and also topological transitions, actually. I mean, it's not trivial also from a mathematical point of view to make a donut out of a sphere, for example, right? So I'm very glad that you will give this masterclass, which is today, tomorrow, and on Wednesday. The general setup is that we have lectures in the morning, yeah. So, and then we have a lunch break here with sandwiches, and then there's a more hands-on tutorial kind of session in the afternoon. Just for me to get a little bit of feedback where you are all from. I think it's always nice. So are you all from UU or affiliated with UU? No, so there are also some, is that the Amsterdam crowd?
I am from Rome.
From Rome?
Yeah.
You came for the masterclass.
No, I'm visiting in the economic geography department.
Okay.
Also, they suggest me to attend the course.
And you?
Yeah, I'm from Wageningen.
Wageningen.
Yes.
Okay, good. Okay, thank you.
Thank you so much, Florian. Thanks everybody for being here. So I'm gonna, the sort of general theme, of course, of the masterclass is complex spatial networks in biology and beyond. And I've organized it, by kind of today going through what do I mean by a complex spatial network? What are some of the characteristics? How do they differ from the typical relational or social networks that people have thought about in network science? What work is out there on complex spatial networks? And in the next 2 days to look at sort of a little bit deeper dive into some specific kinds of complex spatial networks one encounters in biology. So tomorrow concentrating on sort of distribution and flow networks. So think, vasculature, Florian mentioned the brain vasculature. Since I left Rockefeller, I've done some work on liver and pancreatic vasculature and other networks in those systems. And so giving some background. So actually each of the 3 days I've structured in a way to give sort of general background and then some kind of more current researchy topics, mostly stuff that I had something to do with because I know it the best, but that's the sort of general theme for the 3 days. And then the third day, I thought to talk a little bit about mechanical networks, which then connects to the shape programmability stuff that Florian mentioned as well when he was giving me the very kind introduction. So it should be a pretty broad survey. If at any time you have any questions at all, don't be afraid to pipe up. I will be using both slides on the Blackboard, probably like a slide session, a Blackboard session, and a slide session each day, something like that. You know, I was advised not to go back and forth too much, just from difficulty of the Zoom and the camera and everything, and that's entirely fair. But it's also, you know, easier on you guys to have one thing to focus on. Okay, so I thought since this is the beginning of the whole 3 days, I should give you a little motivation. Why should we care about spatial networks, right? And so I used this slide actually go back in my Rockefeller days, my talk in Rockefeller. I was digging through it and I found this, oh, this is nice, I should maybe consider this. So one of the things that's really nice and really important about spatial networks is that it brings together complex topology in principle and complex geometry, right? So you have some geometrical embedding of your network, and it's potentially a very complicated network. And so here, you know, you could never understand New York City. But if you have one hope of understanding New York City, you could start by like understanding all the roadways and maybe all the subways, right? This is the subway map on the left. The subway map, this is a very complicated topological network, right? There's lots of interchanges, there's lots of loops that you could make. When I say complex network, that's a very, generally speaking, complex network is a wishy-washy term, right? It doesn't actually have a real meaning. It just means a big complicated network, right? When I say complex network, I'm gonna mean cyclic. I'm gonna mean that it has cycles in it. It's not a tree, right? So that for the purposes of this course, I'm gonna mean that's what complex networks means. But outside of the context of this course, that may not be the case, right? And of course, there are the limiting cases of very small networks with like one cycle, like 3 nodes and a little triangle. It's not really a complex network because it's boring, it's tiny. But under my current definition, I would still call it a complex network because it has a cycle, not a tree. And the Manhattan subway map is like this. But you know, if you just know the Manhattan subway map by heart, you still would never claim to understand New York City, right? You need to know the geometry too. You need to know the sort of local, you know, the curves, like there's the Chrysler building and much of, you know, the sort of Midtown Manhattan in that picture. But you know, topology is sort of, there's this glib statement that topology is global and geometry is local. And you need both things to understand, fully understand something that lives in space. That global local thing we'll come back to in a minute. Okay, but what about function? So why complex spatial network? So here, these are a couple of really beautiful movies courtesy of Elena Catafori from Penn, that I worked with at Rockefeller and then a little bit since then. So this is fluorescent dye injected into the base of a ginkgo leaf. This is ginkgo biloba at the stem. And so you can see, and with a hole punched out, right? And so you can see ginkgo biloba is not a complex network, right? The venation pattern in the leaf is tree-like, right? It's just branching, branching, and branching with no reconnections. And you know, if you haven't looked how many people have looked very closely at a tree leaf before? I mean, how many of you think that this is the normal thing, the normal situation? The tree leaves are tree-like. It's a tree leaf, it must be tree-like. Question?
No, I was just thinking.
You're saying,
yeah, I think yes.
This is the normal thing, yeah. Any other thoughts? Yeah. So this was the normal thing.
Is that actually, I think it's a needle tree, right? Ginkgo?
Ginkgo?
Have you ever heard about this? I think it's a needle.
What do you mean by needle?
Like a pine, you know, pines have needles and there are 2 classes, there are leaf trees and there are needle trees. And I think this one is a straight one.
This is technically like gymnosperm.
Yeah.
Which is related to the needle. The pine trees are from that clade, I think.
Okay.
But it's one that actually developed a broad leaf instead of a needle, and it had this. So now, yeah, up until about 150-ish million years ago, we'll see the plot in a second, I don't remember, that was normal, but actually the new normal, 150 million years ago, this is a lemon leaf, but actually, you know, pick your random leaf. You look at almost any leaf from any tree, it's overwhelmingly likely to look like this, actually, look very closely. And so here, of course, you have a very complex, very, you know, reticulated, robust, sort of backfilling network. And the sort of the advantage is very obvious. At least one of the advantages is very obvious, right? So you punched a hole into the system. Like imagine that you have, come on in, all good, can see. you punch a hole into the leaf. Imagine that you have some insect, taking a big bite out of the leaf. This happens all the time. For the ginkgo, you take a bite out of the leaf and then you lose the whole wedge downstream of the leaf, right? It's not robust to damage. Whereas the lemon leaf, it changes the timing of things, right? Like, in fact, we played the movie, proximal to the bite or the hole, right? You get sort of slower irrigation than you would have if it weren't there, but you still get irrigation. You still don't lose the ability to reroute. And so actually, this sort of reticulated structure in biological networks is actually very common. Not just leaves, but in other cases. Okay, so as I was alluding to a second ago, this sort of evolutionary adaptation. Just for a second. So these are all the gymnosperms. Many of the clades went extinct. A few of them survived. Ginkgo is, one of them. Ginkgo was actually, maybe 100, 200 years ago, was down to, essentially one grove in China. Like there was a plant, and then because of the fact that it was, prized for actually reasons that I don't remember off the top of my head, but it was cultivated from there, and it sort of has, been protected by us more than anything else. But yeah, when you look at the width of the plot is the biodiversity, and so the angiosperms are just blown up, once they appeared and sort of outcompeting everything else. And this is, mid-Jurassic, so I'm going to stand by my 150 million years-ish estimate. Just staying with the plant theme, right? So this, the leaf example is very two-dimensional, but really, you can have really important effects from, embedded spatial networks in 3D as well. Does anybody know what's special about the aspen tree? Have you heard of an aspen tree? Some of you are nodding. So it's a kind of deciduous tree that's often grows in mountainous regions. One of the nice things that's really nice about aspens is that, like, when in the autumn, when they change colors, you can tell where aspens are because the grove of them all are turning the exact same color at the exact same time. Right? That's why this is all aspen trees. They're all exactly the same shade of yellow at the same time. Right? And that's not a mistake. It's because that's not a few hundred trees. That's one organism. Right? They have a completely shared interconnected root system. That's genetically one clone. It's one thing. That's one being, one plant being. And so the timing, right, of the response to winter and the leaf change onset, and all that change, is not independently decided by some complex, sort of set of feedback, mechanisms as they are in individual trees. It's decided by a complex set of shared feedback mechanisms that come to one really, consensus decision. But the point is that the roots underneath are connected in complex, it's a complex, loopy 3D connection of structures, of which there's not so much good data on. It's a huge network. You would have to actually, dig up the entire grove to actually get the data, and or do a huge amount of very complicated, presumably sonar work that doesn't, the resolution is terrible, right? So you actually can't see because the connections get very small. So it's a little bit crazy. It's something so big has something so difficult to get data on. Okay, and so I told you I wanted to come back, I wanted to loop back. So this philosophically, I would say, the focus of the MasterClass is on these complex spatial networks, but like kind of philosophically, the thing that really excites me about research and about the questions that I want to ask are sort of any place where geometry and topology both matter together in sort of surprising ways for biophysical systems. And so for sure, complex spatial network is one of those things, but just to give you a little philosophical advertisement, let's say. But it's important in order to make that philosophical advertisement, it's important to understand what one means by geometry and topology and why they're important and what even they are. Right. And of course, I think I was told this would be a very broad audience. So of course, there's a precise mathematical definitions of both of these things, but I'm not interested in that right now. now. I'm interested in the concept. Like, what is the conceptual point of talking about geometry versus topology? How are they different? How are they related? So one thing that they're related by is that they're both about shape, right? You want to describe the shape of something, right? If you want to describe it completely, then you need to say something about both geometry and topology. So geometry being, again, this glib local thing, and topology being, again, this glib global thing. So what does that mean? So what that really means is that geometry is the continuous part of the shape, right? It's about derivatives on the shape. It's about differential geometry. It's about rates of change of properties on the shape, right? And those rates of change have to make sense, right? They can't just jump. They can't just do crazy things, right? Whereas topology is everything that's discrete about the shape, right? All the quantities of topology jump discontinuously. None of them are continuous. They all have some discontinuous filtration, or we're mapping onto the integers or something, right? Like, you know, there's no sense of differential changes. There's no sense of continuity even. And so just as an example, right, you have, you know, maybe some Gaussian bump in space, and you can sort of calculate curvatures, which is the second derivative of the position. You know, the first derivative is the tangent vector, which, you know, what the tangent plane is, and the second derivative is the curvature. But you have, you know, sort of continuous locations in space. You can perform derivatives, and, you know, things can get very complicated, but, you know, it's always continuous, and you can always have, you know, you construct operators, you know, tensors and higher order derivatives, and, you know, the Riemann tensor that tells you about, you know, whether or not you're properly commuting the proper curvature partial derivative. You know, all of these things, right? There's a lot of beautiful mathematics, but it's all based on the premise that you can take derivatives locally, you know, and that means that you have to have things that are continuous and have continuous rates of change. Topology, on the other hand, right, like it either is a coffee cup or is a donut, or is not a coffee cup, or is not a donut. Like, you can't continuously be a coffee cup and then continuously be a donut, right? You have to make a discontinuous break, right? You have to be, it's about counting, right? You're counting the number of holes in the surface, right? You're counting the number of connected components of the space that you define. Right, and you can do dumb things like this, where you can put, oh, you know, I circle topology, which means I love topology because the heart and the circle are, you know, they're the same thing topologically, right? Okay, so wait, so let me go back. I think I don't have the slides, so I'm going to say it out loud. All right, so topology, in a way, right? Topology is the thing that is left when you take out all the continuous stuff. Right, you say all these continuous things, that's geometry. And so if I can make a continuous transformation between two states, topologically speaking, I'm just going to say that they're the same, right? And the surprising thing is that that's just not everything, right? Like, you can take away continuous changes, and yet there are still then states that are different, right? Because you are modding out by the continuous changes, then the difference between the states by construction cannot be continuous, right? You have to have discrete jumps. And so, you know, number of holes in the system, number of connected components in the system, these are basic examples, but, you know, you can do whatever sort of topological index you like, you know, whatever higher order homology or cohomology group or whatever else, you know, they're all, you know, indexed by discontinuous objects, by the dimensionality of the space, for example, which is the integer. And so these are the things that are left when you identify equivalence classes out of the differential continuous things. Okay, and just to make a point that's going to come back on Wednesday a little bit, but I'll do it kind of quickly. So we've been talking so far about topology in the sense of the real shapes, the real, you know, real surface, or the topology of a real embedded, you know, vascular network or whatever, or the subway network in New York. But you can also think about the topology of an abstract space, right? So you can sort of, you know, you can assign, you know, you can make a map onto a space that sort of captures all the information about a possible set of rotations, for example. So I realize now that I intended to have a prop. I'm going to dump chalk everywhere. Maybe I can use, not good. Maybe there's some deeper meaning to this, using this sign in this way. So Please feel free to take this with you. So I want you to imagine the rotation space as being indexed by, in 3D, right? You have an axis of rotation, right? And so that axis of rotation can point, anywhere you like on the surface of a sphere, right? So that's this y drew a sphere here. But notice that it's not quite that you have a sphere, you have, if the axis of rotation points like this, or it points like that, right? That's the same rotation just with a minus sign, right? So you come in, you rotate by, some amount around this axis, like this, is the same as rotating, the negative amount on that axis, because you just split the axis. Yeah. And then we're going to index how much you rotate by the distance out from the origin here, okay? And so what that means is, let's imagine that I'm going to just go straight up, right? So I'll put my theta straight up, and I go up, and then it's the same after I go through pi, it's suddenly the same as going through pi going the other way. So I come down to the bottom of the sphere, and then I go back to the middle, right? So one complete rotation like this, that would be going straight up, and then coming out the North Pole, and then reappearing at the South Pole, and then going straight back to the middle, right? And so then that should be the identity, right? Right? Everybody agrees that's the identity? Being back at 0 angle. Okay, so now here's my sign. Please feel free to take it with you. Right, I'm going to rotate the sign by 2 pi. Now the sign is back to where it was. This is correct, right? It was the identity. But notice that my arm is feeling a lot of stresses in it. The ambient space, which is codified by that curve, right, that I took through the space on the sphere, the ambient space is not back where it started, right? And what this means is that curve in the topological, the topology of the transformation space is such that you make one complete loop around the system, and then that curve is not equivalent to not going anywhere, right? It's not contractible. Like the homotopy is not trivial, right? And so now, usually when I show this, by the way, is due to Dirac from the 50s, this little trick. So if you do this, like oftentimes when I did this the first time I did this in Dresden, one of the directors of the CBG like shouted out, "Don't keep going, don't hurt yourself." And of course the trick is that you do keep going, right? So I went that way, and so I'm going to keep going, and I'm back now. I went around twice, and now my arm is relaxed, and it's exactly the way it was before, and the sign is again back to where it started, right? So please feel free to take that with you. So why did that happen? So it happened because of topology. It happened because if you make 2 complete loops around that space, that is equivalent, it turns out, to, it is contractible back down to a point. And so it's a subtle thing about projective spaces. We can talk about it later if you're interested, maybe in the tutorial session briefly. But just the point that topology really matters for, the geometry of topology also really matters for real objects, but also for transformations and transformation spaces. Okay, there's really strong consequences on the stress that is felt by the thing that's performing the transformation. And then again, maybe it's just as not so important for today, but to flag it for Wednesday, a much more simple space of a 2D vector, right? So now your order parameter space. This is the difference between transformation and order parameter. Order parameter, again, conceptually, is you have some space, and then at every location in that space, you assign a property. And the property is indexed by an order parameter, which can be a vector, or a tensor, or a number, or a set of numbers, or a function, or whatever. But whatever the degrees of freedom of that order parameter are, that thing that you're assigning at every point in space, that can be described as well by a topological space. And so a vector in 2D can just point at any direction along the circle. Now the top and the bottom are not equivalent because a vector pointing up and a vector pointing down are different. And the consequence of that very, very simple statement is the entire structure of 2D topological defects and vector fields have to look like that, right? They have to be integer charged because it corresponds to the winding number of the vector, right? And the idea is that when you go around, you have make some path in your space where you're indexing your order parameters. you go around some closed path that corresponds to like everywhere on that path, you know, if you're looking at your order parameter space, your vector must be pointing somewhere on that circle, right? So you go around some closed path, let's say over here, right? Then the vector will like kind of tip, it'll tip that way and then come back, right? And so it won't wind around the circle. But if I do a path that goes around the topological defect, then when you're looking at what happens on the circle, your vector will go all the way around and make a complete winding number, right? So you can't undo that. And so the fact that this space has winding numbers means that there must be some ways to sort of access them in the real space that you paint with the order parameter, and that's what topological defects are, if you've heard about topological defects. So, okay, what does this have to do with networks? Again, so this is a little bit of philosophizing at the beginning of the 3 days. Wow, this is taking way more time than I thought it would. So strategically speaking, right, when you want to bring together topology and geometry, right, one can think about like this is a very coarse way of dividing up all of like, you know, biophysical or physical problems into topology, you know, placing them somewhere on these sort of paired axes of topological and geometric complexity, right? And so I would argue that there's tons of really interesting open problems in mesoscale tissue biology and also organellar biology and organ biology that sit out in that middle region where there's, you know, high complexity of both types. And we just don't have the tools or the know-how to attack them. And so what does one do? Well, you start from problems that are topologically complex or geometrically complex, and you try to build in, right? And so there's been a lot of progress in the last 50 to 100 years of like steadily going out along the two axes. And you know, and not to say there hasn't been anything moving in towards the middle, but there's been a lot less. And so, you know, one can think about, you know, all these different ways to try to do this. And the point of the course is to mostly focus on, you know, this side of the arrow, right? And new approaches to modeling and characterizing spatial networks, complex spatial networks. But we are going to make contact with the other arrow a little bit on Wednesday through the mechanical networks as well. Okay, any questions so far? No? Okay, good. Okay, so now Blackboard interlude time. And so I think we have a break in about 20 minutes, is that right?
Did we start 10, right?
We started at 10, yeah.
Yeah, so 15 to 20 minutes, yeah.
Okay. Okay, so what I wanted to do with the board was to go over likely. Oh, is the camera all good? Yeah. You know, we don't want to reinvent the wheel, right? If we can avoid it. So let's talk about some more traditional network size metrics for networks. In particular, let's think, you know, I'm going to sort of define some of them for those of you that maybe haven't seen all of them. And then we want to think about, are any of these traditional network size metrics useful or not for the spatial complex networks that we care about? Okay. Okay, so how many of you would say you are familiar with many traditional network science, network sciencey tools? Who knows what a centrality is? A couple people, but not everybody. Okay, good. That's about what I thought. So for those of you that know what a centrality is, you know, you can concentrate mostly on the discussion of whether or not these things can be useful for the spatial parts. Those of you that don't know also learn some traditional network science metrics. Okay, so the order is a little bit screwy, but let's talk. First, the most important one to talk about, I think, greedy. Okay, and I should probably make let's make it network here. 1, 2, 5, 7 edges. Okay, so just again, just so that we're all on the same page, like vocabulary wise, I'm going to call the nodes the different sort of dots that one is connecting, right, in the network. So here, sometimes it's also called vertices. So here we have 7 nodes, right? These are the elements of the system, right? So like in a disease spreading model or something, these are your people, right? In a road network model, these are the the junctional points or the endpoints. Right, so the elements of the system and the edges are the interactions or the pathways, right? So, we have, sometimes the edges are labeled by, just the nodes that, that's M1, that's M5. One needs to introduce another dash or something once you have multiple digit numbers or whatever, but that's the idea. So these are the interactions of the system. In general, the edges can have weights. And then leveraging weights is a nice way to introduce a spatial component to the network. We'll think of that later. But the weights just mean, you attach some extra information to each edge, right? It can be, how much capacity there is to sort of, transmit from one node to another. Or it can be the distance. If you have this spatial embedding here, then an edge weight could be the distance between the nodes, right? It can be in an electrical network, it can be the resistance or the capacitance. Okay, and so the degree, I want to maybe this is the number of connections at a node. Okay, right, so the degree, for example, of what we call node 1 over there is, anybody? 1, yes, good. And the degree of node 2, 4, yep, good. Okay, and so, it's very basic, right? You just look at each node and you see how many neighbors it has, right? And so this looking at degree is also looking, for example, at the mean degree of the entire network, which is what it sounds like. You just, take the mean of all of these degrees. This is like bread and butter traditional network science. They're like, when people talk about, small world networks or scale 3 networks being interesting, objects in network science, the scale 3 thing that exists is that the degree distribution is scale 3. That you can look at the distribution of degrees in your network, and you have some sort of polynomial tail, right, or some exponential tail or something like that. But all of that is more usually, typically, basically worthless in spatial networks. So in spatial networks, having a fat tail or any tail on your degree distribution means that you have nodes of very high numbers of connections, right? And in a spatial network, you only have so many, neighbors that you can reach out to, typically speaking, right? So unless you're doing something funny, like neurons do, I should say. Neurons are funny. So they're spatial, but they do kind of weird things because the axons go like all over the place, crazy. Most other spatial networks, you're sort of constrained in some sense by your neighborhood, right? And so that means that for spatial systems, this is not so good. You typically have fixed or almost fixed degree distributions, right? So for example, right, in a crystal network, right? If you have a perfect order in your system, then every single node has the same degree because they're related, all the neighbors are the same. I mean, I guess, maybe you have some by modality if the crystal structure is a little more complicated, but you get the point. Even if you have, let's say jam system, you look at granular packing, you have some disorder in your system, so some neighbors, some guys, some grains have 6 neighbors and some have 5 and some have 7. Well, fine, but you still have this big peak, 5, 6, and 7, you don't have a big broad distribution. So the degree can give you a little information about the spatial network, but it's not much, so it's not very good. So that's first one.
In how degrees?
Sorry?
In and out degrees?
So at the moment, we're not thinking about directed graphs. So we could, of course, think about directed graphs. And then the degree environment is more complicated there, and particularly if you care about dynamics playing out over your spatial network, then absolutely then the degree matters, right? Because then you can think about questions of, and we're going to come to this in a second, of weakly versus strongly connectedness, which has implications on the sort of functional relationship of different parts of the network with other parts of the network, even in spatial context. So for directed networks, then yes, perhaps. But for undirected networks, which so far we're talking about undirected networks, it's not so useful. But thank you, that's a good question. Okay. One might be interested in the shortest path. Okay. And so this is, given a pair of nodes, what is the shortest number of steps one can take to get them? Right, so in this case, if we're talking about 1 and 7, right? I think I heard 3. 3 is correct. Yeah. Example. Okay, so this again, like if these are just the links and don't have information about the spatial embedding, this is not great. So this is for spatially, I would say this, but then with weight information. Okay, it's okay. Right, it contains some useful information. Right? The shortest path, from one place to another, somewhat meaningful, but in a spatial context, usually you have, again, if you're thinking about dynamics playing out over a network, if you're thinking about the interactions, then the shortest path is going to be the sort of strongest coupling between, two nodes spatially, but you're going to still have couplings that occur from all the other paths, right? Like if you think about, if you have some circuit diagram, it's not allowed to just look at the, shortest path between, 2 points, by resistor or whatever, to then forget about everything else, right? Like you have to, think about who's in parallel with who, and you have to think about all the different possible paths. Like this is much more complicated optimization going on with respect to the structure of the network that tells you how the structure function relationship really works, right? So the shortest paths, I'm not going to say doesn't carry any information, but we would like to do better. Okay. All right, so now we're going to have one that's actually kind of okay, and 2 that we're like, eh. So the clustering coefficient, this is somewhat useful. Okay, this is the all my friends know each other number. Right, so that's the ratio of the number of edges between the neighbors of a point, of a vertex, to the total possible that it could have been, right? So let me say that again, right? So it's the ratio of the number of edges at node I. Right? So the clustering coefficient of node I is the total number of edges between its neighbors, divided by how many there could possibly be, which is the degree. Ki is the degree. Maybe I should write that also sometimes. Sorry to make an invitation here. the degree of node I, choose 2, right? Because that's how many possible edges there could be between all of the neighbors, right? So then choose 2 means it's ki times k minus 1 over 2, okay? Right, so that's the clustering coefficient. So for example, C2, right? So 2 has 1, 2, 3, 4 neighbors, right? And among its neighbors, there's only one edge, right? Yeah, right. So then C2 is 6. And but then C7, right? C7 has 3 neighbors and they all of them know each other, right? So C7 has 1, right? So the clustering coefficient is between 0 and 1. And it tells you, again, it tells you about like, of all of the neighbors, you know, how complete is the graph? So does anyone here know what a complete graph is? Does anybody not know what a complete graph is? Okay. The complete graph, just for completeness, complete graph for completeness. A complete graph is a graph where every possible edge is filled in. So you have some n nodes, and then, you know, among those n nodes, every possible pair of nodes has an edge between them. So you couldn't add in more edges if you wanted. They're all there. Okay, so this does tell you a little bit something about the spatial properties, right? Because it tells you, you know, in particular, like, again, it's not great. It's not as useful as it is for like social networks and for click finding and things like this. But it can tell you, particularly when the spatial network, when you see sort of local differences in the clustering coefficient and know it's spatial, then it can tell you something about the local density. It can tell you something about the spatial structure sort of locally. Another really important thing, typically for spatial networks, that one worries about a little bit and it's certainly defined in the traditional traditional network plans metric is connectedness, right? So, you know, most of the times the way that you define a spatial network usually sort of guarantees that it's connected, or if it has some function, right, then it has to be connected to function properly. Like if you have, you know, think about the leaf venation networks. As soon as a part of it got disconnected, that whole part failed to function, right? So it's only the connected part that matters. And then that leads one to think about, you know, clicks, which are connected sub-networks that are complete, right? So a click, you know, this one, 4, 5, 6, 7 is a 4 click. So these 4 nodes, right? They all are connected to all of each other, right? So you have some hyper-connected parts, hyper-connected. So works. And then one can also think about the giant component. So in percolation problems, even in spatial networks, often it's important to think about the largest connected component and how that behaves, right? So that like as your dynamics or as your growth process, you know, if you're thinking about the development of some of these networks, right, you have some growth process that sort of is connecting things up, and you know that you need to land on a completely connected case. That at early times of development, you know, for example, in the brain vasculature or in the liver networks, we'll talk about some tomorrow, you know, you have, you know, lots of little lumens in the tissue that all start growing out independently, little tubes, and they start connecting up. And then the sort of dynamics of how the largest component develops over time is a better word, you know, that is very important. Right? And so this sort of thinking about the giant component, this is good. Like this is a good, useful thing, in both cases, right? So in thinking about the giant component in, you know, like a viral contagion network or something like this, obviously is very important. Thinking about a giant component in, you know, constructing network models like your Ernst-Röntgen networks or your, you know, small world, Albert kind of thing is also very important. But then, yeah, there's clear ways to interpret this for spatial networks. That's good. Okay, and then I want to I promised we get some centrality. Centrality is catnip for traditional network science. There are literally hundreds of different centrality measures out there. One of the earliest and the most, I would say, frequent is called betweenness centrality. I always forget there's two N's in that. Don't quote me on a second. So betweenness centrality, what does this mean? So this means you look at your network, you look at all pairs of nodes. And then you have the set of all the shortest paths between every pair of nodes. Okay. And then from that, you can look at every node. And so there's an edge version of this and a node version of this. That's matter. For the node version of this, you then look at every node and see how many shortest paths of all possible shortest paths does it participate in. You rank nodes by shortest path participation. Right, so like if you have one nice way to think about this, maybe it's worth even drawing a little different diagram. Let me have a break. Edges. So let's take some imaginary spatial network example. Right. And so now, if I now color, I haven't green yet. If I do this, right, what's going to happen? Well, the nodes in the middle are going to have a really big value of the centrality. And the guys on the edge, of course, are going to have this or even maybe 0 centrality, right? And it's going to be more important the closer you are to the middle, right? What that means is this is rubbish, right? Like this is not telling you anything about the structure. It's just telling you, oh, a boundary exists in your system because you have a real system that lives in real space that has a real boundary, and some of your network is closer to the boundary than other parts. Yay, right? Like I could have calculated that in a way easier way. Right, like has no information. So the information that it has is really not very useful, right? And so, what you want from these things is to learn about, what is the local architecture, and by local I mean, not in the geometry topology sense, but in the kind of neighborhood. I look at a little chunk. If I look at a little chunk, the way I drew this, everything close to the boundary has the same architecture as the stuff that I drew in the middle. It's just that the stuff that's close to the boundary has the boundary. Like the boundary nodes are a little different because their degrees are going to be a little different. And then that just completely swaps out the centrality measures. So the centrality measures, despite their ubiquity, sadly, not so great. And I think we should have a little break now.
Could you detect certain in-between boundaries?
If you don't know you have a boundary and you want to look for one, yes. And we've done that a little bit. We played around with that, and it can work.
You see the edges or something.
Yeah. So I have one of my students right now we have a data set where we know that there is a boundary, but we don't know exactly where it is, and we're using something like this to find it. So it is, there is, if you care about boundary, right, and if you want to get that piece of data to extract that information, then you can use this. But if you care about, if you want to actually use it to discover something about the network architecture itself, vis-a-vis its embedding in space, other than the existence of the boundary, it's not so good. Okay, so any questions? No, okay, then how long is the break? 15 minutes? 15 minutes. So we'll start again at 05. Thanks, guys.
Thank you.
Milling around a little. There's this nice facsimile of a plant here that, you know, if you look at it, it has very, very cyclic that works. Printed on the plastic. So, you know, you can take that for what you like.
Oh, it's plastic.
Yeah, that, I think those plants I think are real, but that one. Okay, so I gave you a bit of a survey of some of the more common traditional network science metrics and sort of argued that, you know, and this should come as no surprise, right? These were built, constructed, you know, conceived of for the purely topological part of things, right? For only thinking about the networks, not thinking about spatial embedding. So shock, right? Like they're not that useful for networks where there's this extra, very specific kind of information that can go along with it. Right? It's not totally useful, but not that useful. I think I over planned for this lecture, so I have some more of the traditional metrics that, you know, people are interested. Maybe we can talk about over lunch or maybe during the tutorial or something. But for now, I'd like to move on because otherwise I'm not going to get to, I think, some of the more fun stuff for today. Okay, so how about purpose-built metrics? Like, how about metrics that were made to compensate for or include both the topological character and the spatial character somehow, right? And so, surprisingly, not that many of them, actually. You know, it's actually something, it's still the sort of people researching spatial networks is still a relatively small subset of, you know, kind of sitting in between biophysics and network science, and there's people like Eleni Cadafori at Penn or Karen Leem in Munich. Barabashi has started thinking about it, but only in the sense of like kind of rope tying sense, not the full embedding stuff. You know, me, you know, there's a small community, you know, and it's interestingly, you know, I was at a conference this past summer that was sort of about looping networks, and a lot of the people that are thinking about this are actually coming at it from like the geology and, you know, people thinking about river networks, thinking like the people that want to try to understand river deltas and things like that. A lot of them coming at it, you know, from a 2D angle. So I'm going to tell you some 2D stuff and some 3D stuff. So one of the nice things that was sort of developed more than 10 years ago now is this cycle coalescence. It was sort of independently developed by two different groups. There's people, you know, Edelsbrunner, and who's one of the sort of principles of personal nomology from way back, but also with Weitz and Price, and then also Cadafori and Magnasco at Rockefeller just before I went there. So the idea here is that you have a network, kind of like, imagine like the leaves, right? So you have a 2D cyclic network, and if you have a 2D cyclic network, you can have a bunch of little degree 1 ends, and the idea is to just ignore them, only care about the cyclic. Right? So you throw away everything that doesn't participate in the cycles, and then what's left, because it's in 2D, what's left is now a tiling of the plane, right? You have your cycles in 2D now make tiles. We got to get rid of the little tree-like bits. What's remaining is tiles, right? And so the idea now is that you have spatial information coming from how the tiles sit in space relative to one another, and then further spatial information coming from, you know, somehow the function or the edge weights. You know, if you think about the leaves, for example, you might want to think about the thickness or the flow or the length, or, you know, you add some weights to the system as well. All right, so now you have the topology, the spatial embedding giving you a tiling and some weight set, right? And then the idea is that you now sequentially remove the weakest link. You are the weakest link, goodbye. Right, if any of you are old like me and remember that terrible show. So you remove the weakest link, and then you sort of keep track of, you know, which you had this green plaquette and this pink plaquette, and the merged into a yellow plaquette. You imagine you have a bunch of tiles, so you represent the tiles with a bunch of dots, and then you remove one edge, and so every time you remove one edge, you merge two tiles, right? You know, every time, right? Like without fail. Unless you cut one of the boundary edges, but you can make rules that you don't cut the boundary edge. And so, yeah, so then you keep track of the way that these come together, and then you just iterate, right? You keep doing this, right? And it gives you this sort of relative, importance ranked spatial nestedness of the cyclic structure of the network, right? And you're codified by a tree. And so, this is this nice, one can essentially map a complex spatial network, onto a tree filtration of the geometric and topological data, which is a sort of a nice index. And then one can think about, for example, there's, characterizing and classifying tree networks is a venerable subject with tons of useful tools, that's been worked on for hundreds of years. One that that I think Eleni and Marcelo used that was particularly nice, and that I also use for the 3D follow-up, is this idea of asymmetry, right? So like if every pair, every pair has a parent, and those two parents, have symmetric parents, it's not that you keep branching off, one side like this, right? Like how many daughter leaves are there on one side versus the other, right? It gives you a measure of asymmetry, right? So you can normalize that by how many total leaves there are. That's all this equations are saying. Which over here, like from this node, it ultimately has two daughter leaves on one side and two daughter leaves on the other side, right? So that's perfectly symmetric. Whereas this node, the red circle node up top, has four daughter leaves to its left and one daughter leaf to its right, and so that's quite asymmetric. And so this is, if you go back and think about what the embedded cyclic structure of the networks would look like to produce graphs like this, then you have this sort of graded strength of the edges in the case it's very asymmetric, and then you have a much more fractal-like self-similar construction in space for the case it's very symmetric. So it's giving you this sort of spatial information about the potential existence of local gradients in the edge weights versus the local self-similar structure. Like the more symmetric, the locally symmetric, the characteristic tree for the cycle coalescences, the more self-similar things are, and the more asymmetric it is, the more it's showing you that there's some gradient information there. Okay, so again, that's for 2D, right? And now I would like to, I didn't quite say this in the title of the masterclass, but I would like to sort of push towards 3D on the things that I'm talking about, because I think 3D is more important and it's also more interesting. And so that's of course a personal opinion. You may differ, that's fine. There are lots of interesting things to do in 2D, but most of my expertise is on pushing the 3D side, so that's what I'm going to talk about. Okay, so in this case, it would be nice to have a similar tool. It would be nice to be able to say, okay, I have a 3D network. I don't just immediately have a tiling, right? Because I'm in 3D. So if I don't have a tiling, what do I do, right? And so it turns out this is hard. So how you want ultimately you still have the sense of tiles. You still have these local regions. So here is a very obvious, clear, what you like to call a tile, right? But then you want to be able to cut an edge and merge, one tile with one other tile, right? But here's this edge. Let's think about this edge as the thing we cut. Well, this edge is incident to actually the tile that goes back into the screen, the tile that goes to the left, the tile that comes out of the screen. I mean, do I somehow merge four different things? And after I've made that cut, now suddenly I have one nice thing about making the cut in 2D is I make a cut, I merge two tiles, and the thing that I have left is a tile, right? Whereas here, I make this cut, and I merge these four things. I have this funny shape that has a self-intersection, the line is sort of doubly ramified. It's not a tile in the sense that the tile started out. It's bleh. Well, that's no good. That's no good. We don't want that. Okay. But like, you may protest. Before, you had a tiling of the plane. You had a 2D plane. You had 2D tiles, right, that you covered the plane with. And now you're telling me you want 3D and you're trying to use these 2D tiles. Well, that's nonsense. You need to use 3D, cellular structures. You need to have a tessellation of space by sort of volumetric elements, right? And so that seemed promising, right? Like in the nice graph on the left that I've shown, that would work great, right? But there's two problems with that. So one is that because we're talking about networks here, that means you need to cut, if you have a volumetric structure and you want to merge it, you need to cut the face, right? But all the information from the network lives on the edges, not the face. So you need to come up with some meaningful rule that would, push forward the information from the edges to the face. But even more fundamentally, you're not guaranteed to have a cellular structure at all, right? So if you have a network in 3D, it can happen generically, and actually does happen quite frequently, that distant parts of the network can weave through other parts of the network. And so as soon as you have that weaving, then you no longer have a tessellation, right? Because if you try to sort of fill in all the cellular structures that you get naturally, a bunch of them would be overlapping, there would be regions of space that overlap themselves in other places, and you would be unable to perform any of the tiling tricks. Okay, and so, so one idea one could do is say, all right, well, we have the information about the embedding in the real space. Maybe we can map the system onto some other space and retain the embedding information somehow as some index, right? And so to just kind of run through a little refresher on classical graph theory, we already talked about complete graphs, but just to, I have a slide on it anyway. So, you can draw, the graph representation is any way of drawing the topology of a graph that you like, right? So here, this complete graph on 4 vertices, I drew, this is the usual way people draw K4. I could draw it with wiggly lines, right? That would be a different representation. As long as it has the topology, it's the graph, right? Because again, geometry doesn't matter for just purely the pure topology side of things. Okay, and now, but there's this concept of planarity. So this is a concept that people do think about a little bit in traditional network science that is super useful for spatial networks. So this concept of planarity is when you apply it to a representation, it asks, okay, does the graph that I drew, does it have any crossings of edges, right, that don't, the edges are crossing away from a vertex or a node. Right, so here, right, this has a crossing. So if it has a crossing, it's called non-planar, right? Now, there's a separate, more useful way to think about planarity, not on the representations, because of course the representation you choose is somewhat arbitrary and the space of representation is enormous, right? But is it a graph itself is called planar if it is possible to have a representation that has no edge crossings. And so here's K4 again, right? And now, aha, no edge crossings, right? So K4, the complete graph on 4 vertices, you can write in a way that is planar. And notice that a planar graph is a tiling of the plane, right? So this has tiles, right? And so there's this concept of planarity maps really beautifully onto this key concept that we needed for the 2D cycle coalescence, this tiling. Okay, great. So now maybe this is the answer. Maybe now, okay, the problem is that our complicated 3D graph is just in the wrong representation. We need to write it down in a different representation that is planar, right? Oops. Okay, so obviously you're not going to have a property name if everything has the property. Nobody would have ever bothered to give it the name planar if there weren't non-planar graphs. So of course there are graphs that, ooh, the 5 is in the wrong place, like K5, the index got somehow shifted. So even the complete graph on 5 vertices, so a relatively small graph, there's no way that you can write it down without an edge crossing. It's a fun little exercise, to attempt to do this, right? Like you will not be able to do it, but again, it's a fun little exercise. There's also a really famous, like, and very mean a trick where you have 3, like there's like a brain teaser, right? Where you have 3 like houses and then 3 different utility suppliers, like in the water. 3 houses, 3 utility suppliers. And the goal is to connect all the different, you have plumbing and electricity and water and sewage, whatever, right? Or water, sewage, electricity. And the goal is to connect, each house gets, all 3 utilities go to all 3 houses, but you're not allowed to cross, any connections. That's also impossible, right? So that's a bipartite graph on 3 vertices. That's not, yes, not possible.
Maybe a very stupid question. So I'm actually imagining that you have a 3D, in real life, everything is 3D, right?
Yeah.
So imagine that you have a collection of cells, and these cells are sharing surfaces.
Yeah.
So you can do a Voronoi diagram, for example, in 3D. And then you replace every surface by an edge, network edge.
Yeah.
That will give you a network, right?
That
So
will give
with
you
some
network.
transport, I mean, because.
Yeah, if you have cells, yeah, if you have,
Right.
like, if you have cells to begin with, something that's explicitly cells to begin with, then yes, you have a cellular decomposition, and yes, then you can do what you're saying.
Right. So.
But what I'm arguing is if you have a network to begin with, there's no guarantee you have a cellular decomposition,
Correct.
and most of the time you won't.
Because you motivated by sharing surfaces, that's why I was thinking,
Yeah,
why
yeah,
not
yeah.
that in 3D?
That's
All right,
correct.
so that's a straightforward thing.
Yeah,
to do.
if you have something that you know is a cell has a cellular decomposition to begin with,
Right.
then you can do that. But for a network, you don't have that to begin with, and most of the time, actually, it will fail. There may be special cases if the network comes from, you know, if the network pieces are all in between something that sort of guarantees that it's otherwise cellular, then maybe you're okay, and there are cases where it's like that, but yeah, it's also better to actually, you know, kind of try to come up with a more general idea as well, for the cases that it doesn't work. Okay, so anyway, yeah, so we can't just take our complicated 3D network and like squash it into the plane and then make a tiling structure out of it. Like, shouldn't actually be that surprising. That would be a hard thing to do. Right? You project some complicated 3D network into the plane and then resolve all the crossings. Yeah, it's not possible in general. Very quickly, it's not possible. And so is it even, it's worth asking, is it even possible, you know, this is a very abstract question, is it even possible to take a network, you know, a non-spatial network and embed it into three-dimensional space? Like, can we do that without edge crossings? And the answer actually turns out to be yes, right? So the problem is 2D. So any other dimension, any other higher dimension you can always do. There's this classical thing called the book representation. So this is a representation of the classic section. And so the idea is that you take your network and you line up all your nodes in a line, and you think about that line as being the spine of a book. And then you have a page in your book for every edge. And so an edge connects two nodes, and you have a page for every edge. And so in the pages each have a, you know, a separate angle in 3D. And so no edge will ever cross another edge. And you can put in every single edge you want because you can put in as many pages as you want, right? And so you can always take, no matter what the network is, no matter how complicated it is, it can be the internet, right? You can make a 3D embedding of that network with no edge crossings. So the issue for the making the tilings really is about the planarity, about getting into 2D. So getting to 3D is okay, 2D not so much. I should point out, right, that like, this is you can make this embedding, but it's not very useful, right? Like from the point of view of like, you know, a network representation, it's a mess. But it just shows that you can always have a 3D network in embedded, you're properly embedded no edge crossings in 3D space. Okay, and so maybe that gives us an answer, right? So you can do it in 3D, so that means that like maybe you take that book embedding and you chase all the edges with like a little tube, and you know have all the nodes as a little like, you know, a little connector ball. And that means that suddenly not only are you doing it in 3D, you're doing it on a surface, a 2D surface. Right now that surface is a nightmare because you have every single edge has its own little handle. Right? So topologically speaking, like the genus of that surface is enormous. But that suggests a concept called the graph genus, right? So this is a nice sort of higher order concept about graphs and network theory. So every graph, this is fundamental. This is weirdly not about the spatial embedding, but about the topology of the graph. But it's about how the topology of the graph can embed into space. So like it brings in the spatial embedding in a funny backwards way. And so the graph genus is the simplest, topologically speaking, the simplest surface, the genus of the simplest surface that you can embed the graph in without an edge crossing. And so every graph admits, you know, some embedding onto a surface with no edge crossings. And then, of course, you can always make that surface more complicated if you want, but every graph has a simplest surface, right? So the planar graph, the simplest surface is a sphere or the plane. But for K5, for example, right, you can just take a torus, right? So you connect the edges, the top and the bottom are, you know, wrapped one way, the left and right are wrapped the other way. And suddenly, K5, which you could not embed on the plane, you can embed with no edge crossings by allowing yourself to sort of go around the generators of the torus, right, with the cyclic directions. Okay, and so this is now a tiling of a complicated surface. And so, okay, great. So maybe now here's the answer. So we have a complicated 3D graph. You want to map it to a potentially complicated, but not as complicated surface, where it gives us a tiling, and then we perform the same tile coalescence on the surface. Okay? Shoot. Okay? Finding the graph genus is NP hard. And so, spatial networks are complex. Who would have thought? Let's table that for now. Remember that, don't forget, we're going to move on to some other stuff. And at the end, if I time it right, we'll loop back around to this problem that seems like we can't solve. Okay? Because we ran into the NP hard, brick wall. Like in principle, there is a solution, but it's going to be, exponentially difficult to compute and, not worth actually pursuing. Okay, so what else can we think about? All right, so that's complicated, massive picture. This is actually beautiful data taken. I should have had a little attribution out here. This is beautiful data from the Zerial lab at the CBG in Dresden, and also now the Human Technical in Milan. The green are the biocanaliculi, the canalicular networks in a liver lobule, and the purple are the sinusoidal, the blood networks in a liver lobule. Okay, and you get this kind of hole that you see up here is the central vein. So you have a large central vein in each lobule, and then the portal triad, the portal vein, and the bile ducts on the other end. And there's all these capillaries that go between them, and these capillaries are very, very highly cyclic. Both of them, both networks, the blood network and the bile network, are very highly cyclic. So this is a beautiful case of two complex spatial networks with a very important function. And note that these are absolutely separated, different, distinct networks. They do not have edges that flow in one another, right? You do not want bile getting into your blood, right? That is bad news. Okay, so there's very important sort of adaptive reasons to keep the network separate, but they also need to service the same space. And so one question one might ask is trying to understand this sort of mesh of structure. We can start with some more basic questions about, all right, like how are the neighbors? Like you have a branch point, does it kind of reach all of its neighbors? Is the network being formed by sort of some concept of neighborhood or not? And generally speaking, concepts of neighborhood, who is my neighbor, is a very spatially dependent thing. And so if we can sort of connect that to network structures, that's a good start towards making an explicit network structure, as a network metric that includes the complex and the spatial. Okay, and so who is my neighbor? And traditionally speaking, there's three popular ways to determine. You have a point cloud, and you are asked, okay, who are his neighbors of who? The traditional ways to answer that question are the following three things. You can either pick a number and then find the closest four nodes to every node, and you make those connections, right? So this is the graph that you produce from looking at the nearest four neighbors of every node. Okay. Alternately, and probably the most popular one, is to choose some specific distance. And then you take that, you look at that distance away from every node, and you find all the other nodes that are within that distance, and then you connect them up, right? So this is an example here. And then another very popular one is the Delaunay graph, which is the dual to the Voronoi tessellation, which was brought up earlier. So the Delaunay graph is a way to generate a structure that sort of looks kind of more like a tiling. It's triangular. The best way to think about it is that there's a definition based on sort of what triangles have overlapping circles of being inscribed. And that's a perfectly valid mathematical definition. I don't find that it has much conceptual use. The fact that this is the dual of the Voronoi tessellation is much better, I think. But by dual of the Voronoi tessellation, that means that like every node, and then you can see it's kind of faint, but the Voronoi tessellation is actually out here in red. Every node is the center of one of the Voronoi cells, and you draw in an edge, across, for the Voronoi cells that are neighbors, in exactly the same way that you proposed earlier, right, for the 3D cellular structure. So that produces the Delaunay graph. Okay, so these are the traditional ways to assign neighbors. Okay, these are all, you know, you can look at them, these are all quite coarse, right? Like the Delaunay graph is the most complicated of them, the other two are very coarse. Maybe we can do better. Do we need to do better? I would guess that all of you know who is supposed to be the neighbor of who from this point cloud. That's terrible. The other metrics, the other ways to do it don't do any better, right? So for many cases, we do want to do better, right? We want to be able to have a way to sort of assign neighbors that really is sensitive to what we would like to call shape, right? And so there's this actually really clever idea from the 80s in computer vision called the relative neighborhood graph, where you take every pair of points, you draw a line between them, you take that line to be the radius of a circle centered at each of the 2 points, so these 2 orange circles, and then that generates this overlap region of the 2 circles. And then you look in that overlap region, this little loon, and if there are any other nodes from your graph in the loon, then your points A and B are not connected by an edge. And if the loon is empty, then okay, then you draw an edge, right? And so this is great because it's in some sense a little bit scale free, right? Like so you can have there's no like local scale baked into it, right? The scale that came from the the neighbor distance choice. It's also nice because unlike all the other metrics, it solves the problem of occlusion, right? Like if I want to know if I'm neighbors with Florian, I'm sorry, what's your name?
Griffin.
Griffin. So if I want to know if I'm neighbors with Florian and I make my like distance halo to go to the plants by the windows, then I would say, yeah, I'm Florian's neighbor. But Griffin is like directly in between us. That's crazy. Like Griffin is my neighbor and then Griffin is Florian's neighbor, but like he's in the way otherwise. So there should be a way to sort of assign neighborhood that takes account of, occlusion or blocking. And this does that because you look in this intermediate region, right? And you can then, if somebody is in between, then you say, oh, wait, no, these aren't neighbors. So that's good. Now notice, of course, that the cat is not perfect. So it's not perfect, but it's good. Okay, but that's just like a single way to assign neighbors. It would be more useful actually if we had a family of ways that we could then like do scalings with and do like physicist tricks with. And then from later in the 80s, from more computer graphics people, they realized, oh, well, actually you can flex the relative size of this loon in a way that's sort of straightforward. They called it, they gave the parameter the name beta, so these things are called beta skeletons. They've been largely dropped since then because computer graphics went a different direction, and nobody actually picked them up for research, as far as we know. There are some people that have done some stuff with them. I shouldn't say nobody, but they've been underused. So the idea now is that as you make, so the beta equals 2 is the case that we looked at on the last slide. And as you draw beta back closer to 1, you get sort of a smaller, occlusion region. And as you make beta larger than 2, you start, pushing out more and more until as beta goes to infinity, you get like an infinite strip, between the 2 vertices. It generalizes to 3D or arbitrary dimensions, if you like. And then you can sort of sweep through the different values of beta. The 2 is the one I showed you before. 1 is like a little bit over-connected. Under 1 is wildly over-connected. You can go way above 2, and then you start losing connections. And so the idea here then is that you have some point pattern, and this point pattern can come from a network, right? You can have the branch points of a network, and then forget about the network that you have, and then think about those branch points, and then generate a series of networks, and then compare the properties of that series either from specialized spots and then a match to the real thing, or the scaling properties of the networks with beta to tell you about the sort of neighborhoodiness of the structure. So this all seems a little abstract. I understand that. So we have some examples. So first, to point out that as you raise beta, right, every subsequent network you get is a subnetwork of the previous ones, right? And that stopped at beta equals 2. Oops, I thought it kept going. So this goes up to beta equals 2. And now we recover the nice, almost perfect scale lattice with one missing, even with noise. You go above 2, then you start losing those edges. right? So it's also nice that you get a sequence of subnetworks, right? So you only lose edges as you go up in beta, right? So then it can make sense to look at the rate at which you're losing edges, for example. Some of the individual betas are special. The Gabriel graph is a particular special graph that's separately defined. As beta equals 1, the relative neighborhood graph we talked about is separately defined. So long as beta is greater than or equal to 1, all of the graphs are subgraphs of the Delaunay graph. So long as beta is greater than or equal to 1, if you have a 2D set of nodes, they're all planar. Once you go under 1, you start getting non-planar graphs. As beta approaches 0, you get a complete graph. And you're guaranteed to be connected up to beta is equal to 2, and then you generally can start disconnecting the graphs if you go up to 2. So there's sort of lots of special landmarks and properties, throughout this parameter. Just graphically speaking, I think I said all these things already, but the different betas are subgraphs of the Delaunay graph. The beta equals 2, the relative neighborhood graph is a subgraph of the Gabriel graph. The minimum Euclidean spanning tree is also sitting inside of these structures, and so is the nearest neighbor graph, this is graph of this as well. So there's lots of important spatial information is already captured in this family. So it's good. Going back to the theme of like the fact that it's sort of scale free in the way that you look at the occlusion means that you can reconstruct the lattice structure really, really accurately, even if you have some affine transformation to the lattice. So in this case, we sort of, twisted and bent the lattice in lots of crazy ways, and the lattice structure is still completely recovered by a specific value of beta. A really nice thing about this from the point pattern side, again, forgetting about the networks, but just looking at point patterns, is that if you look at the scaling, if you look at this should be a movie, it's not playing. If you look at the scaling of the number of edges, the fraction of the edges, as you increase beta, so you increase the area of the occlusion region, you get this beautiful polynomial, this beautiful power law scaling. So there's this U-1 power in the decrease if the point cloud is produced by independent assignment of the points. Right, so if you have it doesn't matter what the distribution is, what the probability distribution you're pulling from. So for example, this one on top is you're pulling from you have uniform probability to choose a point anywhere on the disk, and you just independently choose points over and over again, right? And the one at the bottom, you have a much higher probability kind of in this kind of funny crescent shape, and then much lower probability farther away from that, and then you independently pull points over and over again. And in both cases, you get the exact same scaling because it was independent. You would independent draw the points, right? So if you're basically if you're creating your point pattern with a Poisson process, then you get this beautiful -1 scaling, and otherwise you get more information. Right? You can tell them apart. And so this is out of order, I think.
I have a question. So you have analyzed then if you have a point pattern, and I construct now these networks for different beta, and then I look at the scaling of the beta, then I know if it's a complete or spatially random point pattern.
Well, it can still be random, but if it's random with history, with knowledge, with memory, right, then the scaling gets moved. It has to be independently random.
Independently, right?
That's the thing that you pick up with this.
Okay, and then
so this is the slide that is actually I think is missing. So we're just showing a couple of examples. Which maybe I can find.
And that works already for a relatively small number of points, or do you need?
What do you mean by relatively small?
Like this on the left, maybe is that?
Yes,
something you could find out. And then like the consequence would be you could identify clusters or spacing, right?
Yeah.
If it's not, okay.
For example, yeah. So I would maybe, I think I would jump to a second. I'm pretty sure I'm missing slides in the show. Yeah. Well, actually, maybe, so there's these extra slides where this is shown for some non-independent cases, but maybe we can talk about that briefly in
Yeah.
the after session. So for bushes in the desert and for the pores in a diatom shell, you can do this through this. Totally random, different cases, but okay. So let's back to the liver network. So I showed you this picture of the liver before. This is what happens now if you take returning to you now using this tool as a network characteristic tool, right? You can then think about this is you can make just take just the blood network, just as an example. This is the actual network extracted from the blood network. This is if you attempt to, you take all the branch points and then you try to do the Delaunay triangulation, you get a little mess. You also, totally lose the shape of the broader shape of the data. And you get this, the Delaunay triangulation is guaranteed to give you something that's convex. But there's two different, beta equals 1 and beta equals 2, both do a better job. The Gabriel graph is clearly too much, but the relative neighborhood graph just by I already looks pretty good. One can do better than just by I. Right, you can match the number of edges. You can take beta, remember, so beta, you have a value where, beta has way too many edges, and you know that the edges, go to 0 as you go to beta goes to infinity, so there's going to be some value of beta where you match the number of edges of the actual network. It turns out for this case, it's around 1.8. And then you can look at the, so we calculated the Jacquard similarity for this, and you can, of course, find individual spots where there's a difference, in the beta, you have an edge here, and you have an edge here. You can just, again, by eye, you can see that in the real network, there's this kind of trend towards almost like a pneumatic ordering and pointing towards the central vein location, which is missed by the beta equals 1.8, which we can reproduce if we flex the occlusion regions. The occlusion region at the moment is isotropic, but one can make it anisotropic and you can recreate this effect. But actually, the Jacquard similarity is very high. So the topological structure but this is very good. And then one can then look at something like the edge length distributions in the network and then the beta skeleton graphs. And one finds, interestingly, in the empirical, blue is the empirical graph, you just have this peak at a certain value of the edge length and then a big fat tail. But for the, and also for the matching edge count graph, you have more or less the same behavior. But some of the other beta graphs have this funny bimodal behavior where there's a peak at a low value that's sort of the similar value to the peak of the real network, and then another peak at a later value, right? And so what's going on with that? And actually, if you really sweep through more and more betas, you really see it, right? Like you have this value that, at high betas, you have this match, but you push it down to lower betas, and suddenly you discover there's this funny second shoulder, this second peak. So what does that mean? Well, if you have a lattice structure that you're reproducing with your beta skeleton, this is exactly what happens, right? Because in a lattice structure, you have the sort of primary lengths get repeated over and over again, like in a cubic lattice, let's say, you have the lattice spacing, but then you also have the diagonals, right? As something that can pop in, and then suddenly you have lots and lots of diagonals. So you get a second peak, from sort of secondary length scale that's repeated in a lattice structure. And so perhaps this is telling us that the organization of the liver has, surprisingly some order to it, some sort of lattice-like order to it. And oh, I don't have that slide either. So sadly, so there's an unpublished data. The Zariel lab had found that these sinusoidal network is composed of these funny cages, right? So there's this sort of sequence of hepatocytes from 3 to 11 in a linear chain that are enclosed by the blood network in sort of caged structures. And so it's not a perfect lattice, but there's definitely much more lattice-like order than you would have thought that, actually is sort of backed up by this, much more abstract sort of, spatial network analysis. And I have a slide with that, but it's somehow, it's in the other talk. It didn't make it into the talk. It is unpublished, so maybe I shouldn't show it. I don't know. I'm telling you. Yeah. Okay. So 15 minutes. Okay. There's another sense in which you can look at this thing and say, all right, well, one of the things that jumps out at you about this picture, right, is that this is two networks sharing the same space and they're both cyclic, right, and they're in 3D. And so that means that something special can happen, and something special is happening all over the place here, right? In 2D, even in a spatial network in 2D, if you have cycles and different networks, then you can't they can't invade each other, right? Like they're tilings, just like we talked about before. You have, each 2D network is going to make some local tiling. You can have like different tilings and they can be kind of interleaved, but you can always partition the space into the area of one set of tiles and the area of the other set of tiles. But in 3 dimensions, right, you can have these cycles of one network and cycles of another network, and they can be linked, right? Like one cycle can pass around or through the other cycle. And it turns out this happens all over the place, right? In the liver, in other organs, this is actually a super common refrain, right? And so this is something that this is a very recent paper that came out that we called this ensnarled networks, or networks where you have multiple cycles topologically linked between 2 different networks. And so, topological linkage is, very venerable, subject. People have been studying this for 100 years. But a metric that to describe how 2 different complex cyclic spatial networks are relatively linked with one another doesn't exist. And so, largely because this data didn't really exist, so there's no reason to sort of push at it. But now, we have this data revolution in biology, even at the mesoscales and the tissue scales. And so now this becomes relevant. And now, one can start thinking about how to do this. And so the idea, who knows what the linking number is? Okay, so these are some examples of different kinds of topological links between 2 curves. The simplest one, the one that I showed with my fingers, is the Hopf link, where you have one curve, one time linked with another curve. But you can have other sort of multiply linked structures where you kind of wind over the one curve winds around the other curve multiple times as it goes. One sort of, if you're able to project things cleanly into the plane and sort of resolve crossings from your projection, you can calculate the linking number in this very easy way, where you choose a, one of you color the 2 curves, and you pick one of the curves to be, your red curve, one of your blue curve, and then you have these 4 different possible cases essentially where the ordering can go, you can go, if you point your red curve always up, you think of that as going up, then your blue curve is either on top going to the right, on the bottom going to the right, on the bottom going to the left, or on the top going to the left. Your red curve, and you assign either +1 or -1 to each of these. So these 2 are +1, these 2 are -1. And then you go through all the, on your link, you go through all the possibilities. So here you would go through, you should just check these 2, they're each +1, and you divide by 2 in the formula. And that's the linking number, right? So it turns out that this little flipping of looking at the crossings gives you a way to calculate, how much they're linked. But basically the idea is just this simple link, it should have a value of +1. If you flip the directionality, you should have -1. So the -1 +1 doesn't really matter, just the norm. But, if in complicated data sets, actually doing this projection and being able to resolve the crossings is not so reliable. So it's better to do something in 3D, which is the Gauss linking integral, where this is, those of you that are physicists might be familiar with this from Maxwell's laws, where it appears as the Briggs-Sawyer law as well. Basically, it's this nice integral that looks like a nice continuous thing, but actually encodes some topological data, right? So that this integral is 0 if the curves are not linked, and it is equal to the linking number if the curves are linked, depending on what linking number it is. Okay, so this is a way to calculate linking numbers. We want to be able to think about linking numbers of relative cycles, right? So how many, there's a natural question that one can ask. We're talking about these complex cyclic networks. How many cycles are there in a network? Or you have some complex cyclic network with tons of cycles. What do I mean by tons of cycles? Like, how many cycles are there in this picture? How many cycles are there in this picture? Who says what? Any guesses? Infinite cycles, that's an answer. That's in some sense the correct answer, actually. But there's another good answer. Anybody else see another good answer besides infinite? No, just infinite.
9.
Nine is another good answer, right?
14.
14, maybe, where's the 14 coming from? Oops,
So,
sorry, go ahead.
so 9 individual circles,
Yeah,
1 big circle,
yeah,
and 4 square circles, but I can recognize rectangulars.
yeah, but there's rectangles and there's like Tetris shapes and there's, so you could do that, but it would never be bigger than 14. So the possible answers are basically infinite, 9, or some large number that I'm also not sure what it is, but something like 100-ish, right? And oops, so I'm hitting the buttons here as I talk. And so, okay, infinite is, you know, in some sense correct, because you can go, you know, here or here around here twice, and you all have winding numbers, and you can go as many times as you like. You know, also the 100-ish is some sense correct, but we would like to work with the smallest number that we can. And so 9 is actually the one that we want to work with. Why 9? Well, so here there's 9 tiles, right? So there's 9 tiles, and actually, if you put a really simple vector space structure over the system, you can take those 9 tiles and use them to build all the other possible loops, all the other possible networks, all the other possible cycles. And so it turns out that that's a vector space. It's called the septal space. It's also isomorphic to the first homology group, if you know or care about that. But the point is, it's a vector space, a simple vector space. And so you can generate, because it's a vector space, it means it has a basis set, right? And so that basis set is some minimal set of vectors in the space that allow you to construct every element in the set. For a tiling, you know, the tiles are but they're not the only basis set, but they're a really nice basis set. And so here this has a basis of 9 different vectors, so it's a 9-dimensional vector space. But more generally, really going too fast. There we go. We're generally one can make a spanning tree, right, of the network. So that's a subtree that visits every vertex. And then by, you know, even if I don't have a nice tiling, but I can just take the tiles. If I have a spanning tree, then every edge that I have a spanning tree plus one edge will always just make one cycle, right? And then I can take that edge away and then do a different edge, right? And that will make a cycle. And that cycle will be distinct to the one that I made before. And if I do that for every edge that's not in the spanning tree, sort of separately, then the set of cycles that I create are a basis set for the cycle space. You can build every possible cycle out of those cycles. Okay? Now, one can ask, like here, these red circles are the basis set generated by this spanning tree. Notice that this is like mostly just the tiles, but there's one and a bigger one that, you know, fine, it's okay, it still works. One can ask, is there something special about the tiles with respect to it being a basis set? And maybe kind of hold that in the back of your mind for a moment. And let's go back to talking about this installment, right? So what we want to do is we want to think about who is linked with who. And we don't want to, you know, check an infinite number of cycles against an infinite number of cycles in the two networks. And we'd rather not check, you know, 100 plus with 100 plus. We'd rather do 9 and 9, right? Or however many basis set is. And so what we can do is we can generate a basis set for each network. We can calculate the linking number whichever way is more convenient for the network that we have, either with just the simple crossing rules or with the Gauss linking integral. And then we can do the dumbest possible thing and just write a table, right? And say, okay, like here, you know, in this position, we have cycle 1 from the first network and cycle 2 from the second network, and those two cycles are linked with linking number 1, right? And we write a 1 there. And this corresponds to these two networks. And so this is just a dumb table. It's not covariant. It's not a tensor. It doesn't transform like a tensor. It doesn't transform at all. It's not really great because it depends on this totally arbitrary choice of basis that we made when we chose an arbitrary spanning tree. But, you can factor out the arbitrary information by the mesh matrices, which relate the cycle space with the edge space, right? So for every cycle, it has certain, you know, it contains a certain distinct number of edges, an identity of set of edges. And so the mesh matrices allow you to pull back the structure off onto the edge space where suddenly you factor out the arbitrariness and you create a covariance structure. And so you have this nice tensor, independent of the basis. Because the number of edges in each network is not necessarily the same, it's nice to work with square matrices, so it's nice to square this. And there's sort of two different possible two-sided squares, one corresponding to information in the space of the first network and one in the space of the second network about how they're installed. And even already here, before you do anything fancy at all, right? Like if you just look at these lambda matrices, which we call the edge priority matrices, just if you look at the diagonals, like not even doing anything spectral, nothing with eigenvalue, just the diagonals, right? These are now the sort of the darkness of the color encodes the size, the magnitude of the diagonal corresponding to it, and it actually lights up for the edges that are important for the installment, right? And it turns out that just the diagonals of this object give you a nice sort of greedy algorithm for the fastest way to disconnect ensnarled networks. We've done some preliminary characterization of the liver. We already find some interesting signals just to sort of indicate like, hey, this kind of is cool. So for example, in space, so this zonation index is sort of as chi is increasing as you move away from the central vein. And the minimum cut, this one on top, is the minimum number of cuts one needs to disconnect the installment of the two structures. And so there's this interesting, like you move away in chi, you have some noise, but it's relatively high, and then suddenly it drops down almost 50%, about halfway to the portal vein. So this is chi goes here from the central vein to the portal vein. So there's this really interesting sort of geometric topological structure of the installment that's occurring in the liver tissue that we don't know what the purpose is yet. It's again super preliminary. And then also now this is in time, so this is at postnatal weeks. And we did actually I had a master's student where we did some spectral analysis of this operator we built. And the eigenvalues is, the number of non-zero eigenvalues is also a nice readout of the sort of degree of installment. And so here in the frequency, the number of non-zero eigenvalues after the birth of the mice in this case, you see that the networks are getting less and less ensnarled, and then suddenly the installment picks way up. And at first we thought, okay, this is a really cool signal, but we don't know what it means. And then we were showing it to a sort of a mouse liver, a liver organoid expert, and she was like, oh, well, yeah, but that's when they start eating, right? And that's when they start, they're on milk, mother's milk, and then postnatal week 4, they start being weaned, they start eating actual food. What's the function of the liver? Well, the function of the liver is to do a lot of filtration of impurities in the blood and bile acids that are required to break down fats. That you suddenly have this sort of very specific profile of the nutrients you're getting from the mother's milk, and suddenly you're eating whatever you're eating, right? Like, and so maybe this is the idea is perhaps this is a functional response to the change in diet. We don't know. Again, it's super preliminary. But wait, we have 3 minutes left. What about those cycle embeddings? Right. So something we talked about actually provides an answer, and it's this. Right, so what I asked a minute ago to hold in your heads, what is the difference? What is the connection between these basis vectors and the tiles? Right, so what is the connection between the basis vectors and the tiles? There's tons of different basis vectors. Like this one not being too big is kind of screaming out at you, right? Like a tiling constitutes a basis set that is in some sense minimal, that uses the fewest number of edges, right, or has the least distance in the edges. Right, like because this long one, we can make it more parsimonious if we just drew it back onto the tile, right? And so maybe there's a way to think about, this. Remember what we wanted to do? We wanted to map our 3D or potentially 3D complicated, complex network onto a 2D surface, right? And where we didn't know what the genus was necessarily. And so how could this work if we have a minimal basis? So here's a minimal basis wrapped around a torus, right? So this is a simple case, right? And notice that your minimal basis is going to be composed of lots of things with a very low number of edges, like all these squares have 4 edges. But because it's a basis set, you need to be able to get every cycle of the system. So that's going to include the cycles that go across the the periodic boundaries, like across the long, the handles of the torus. And the idea would be that you find a general, you generate this minimal basis, and then you assume that now, okay, my minimal basis, I'm gonna have tiles of my surface structure, and I'm gonna have these longer things, right, that are gonna occasionally pop in, that are gonna be going around topological directions. And I'm gonna make the sort of statistical assumption that every time, I want to try to, I want to do this merger, I want to make this filtration. So I choose an edge to cut, and if that edge only has 2 cycles that are, next to it, then, okay, those must have been tiles, so I just go ahead and cut them. And if it has more than 2, right, then I'm going to say, oh, well, the only way it can have more than 2, how can it have more than 2? It can have more than 2 if it's also sitting on one of these topological cycles, on one of these cycles that go around the periodic boundaries. So if it has more than 2, then there must be 2 tiles that I'm assuming are small, and it has one of these longer things. So I just take the smaller 2 and I merge those, and I forget about it, right? And so this gives you a statistical way. So statistically speaking, you can be making errors occasionally in those choices, but the choices are well defined, and the errors turn out, I'm not showing it here, but the errors turn out are very strongly bounded. And so if you do this with a large enough network, you statistically generate the right structure, right? Like you don't explicitly solve for the graph genus problem anymore, right? Because you're doing it statistically and there's an error rate. But because it's statistical with an error rate, it's not the identical NP-hard problem anymore. It's a different problem that's not NP-hard. And so you can do it. And so, yeah, so by kind of tying together these sort of very disparate parts of graph theory, it turns out you can do some fun stuff with spatial networks, and we are 3 minutes over time. And if any of you are looking for postdocs in the future, there's a nice postdoc who I'll advertise this every day at the end as well. There's this nice sort of independent postdoc fellowship that we have in Dresden. So if you're interested in any of the stuff or any of the sort of biophysics-y, quantitative biology, applied math biology stuff, and are coming up on thinking about postdocs, then do think about the postdoc fellows program. And I'm happy to take questions. Thank you.
Thank you. Questions?
Yeah, can I ask another question?
Of course.
So the way you had mapped from beta, the actual, let's say, observed networks in reality to beta, the data network, the beta parameter, then you showed that you had by tuning that beta parameter, you basically can regenerate the empirical network.
Yeah, we do a good job of regenerating. It's not perfect.
Yeah. My question is, what do you exactly learn out of this exercise?
That's a good question. So I actually think I think the fact that you can kind of regenerate the network at a certain value of beta is nice, but it's actually the less useful part of this. I think the more useful part of this, it comes from things like this example, where you actually use the whole spectrum of the betas to find a hidden structure that is like sitting at values of beta that are not where the real network is. But in a real network, and the value of beta that best reproduces it, right? You just have this monotonic distribution of the edge lengths, and you don't see any signal of this lattice-like, this sort of hidden lattice-like structure. But the fact that you have this sort of related family, the spectra of families that has this appear, and I didn't show the sort of negative control here, but if you have a completely distorted lattice to the point where the noise overwhelms it, or if you have a Poisson process or something like that, you don't get that, right? You get just a single, distribution with a single scaling, right? The single tail. So I would argue that you want to be able to, the fact that there is a value of the beta that does a good job of reproducing the network tells you that the rest of the tools can be meaningful, but on its own, it's not that useful. What's useful is then look at the properties of the family of betas. You look at the scalings, look at other properties or other examples that we can talk about. But I would argue that that's the thing. You do that as the first step to make sure, okay, thinking about just the branch points is enough to encode most of the information. And then you can look at the spectrum at the family of the beta networks and look at their scalings as different ways. To sort of to deduce things about the properties of the real network.
Yeah, don't get me wrong, this is very beautiful, but I'm just wondering, I mean, what kind of practical consequences would this actually have? That's basically what I mean.
Yeah, it's an important question. I mean, again, my answer would be something like this, where this would tell you to look for an underlying lattice-like structure where, you don't see it right away. And where it would be a surprise, and it is there, we can tell. Other questions?